Hi there, my name is Lindsay Roberts, and I will be presenting on chemoreception and how that is a process that applies to olfaction as well as gustation. Now, when we talk about chemoreception, it is a process in which a chemical substance binds to a sensory receptor and then is essentially transmitted to the neuron to be processed and trigger those action potentials. And there's a couple of ways in which chemoreception can be applied to olfaction, sense of smell, as well as sense of taste. In the case of olfaction, the odorant molecules, they bind to olfactory receptors. In terms of our sense of taste, what are called tastants, which are, again, molecules that are dissolved in saliva, they bind with receptors on the taste bud to then be processed eventually by the brain. Now, when we think about chemoreception in olfaction specifically, these odorants are dissolved into the mucus of the nasal cavity. So they basically transduce from a smell to more of a molecule, and they bind to what's called a G protein linked membrane receptor, and in this case it's called GULF. Now, these G-linked protein receptors, they are integral proteins, which we talked about in Chapter 3, and these proteins span the phospholipid bilayer, and they have a couple of different functions. So they allow the cell to recognize and respond to molecules in their environment. So in the case of these olfactory odorants, these are basically reaching that phospholipid bilayer and communicating with these integral proteins. Now, when the odorants reach the phospholipid bilayer and they reach the proteins, the binding of the substance activates an enzyme that converts ATP to what's called cyclic AMP. Now, when this binding occurs, this causes an increase in this CAMP nucleotide. This triggers depolarization, which very frequently talked about so far this unit. This triggers depolarization, and that subsequently causes the opening of ion channels. When these ion channels open, sodium and calcium rush into the cell, and this essentially makes the cell less negative because we're changing the resting membrane potential. This change in the membrane potential triggers that action potential that travels to the next part of the olfactory system called the olfactory bulb. Now, when we talk about chemoreception with our sense of taste, there are a lot of different types of receptors, 4 that we talked about in the text. The first type is what codes for salt, the tastant salt. And this actually, this signal transduction is unclear in human models because most of the research has been based on animal studies, specifically mice. So researchers hypothesize that the transduction is similar, but it's not confirmed. In the case of salt, this particular tastant molecule binds to the receptor cell on the taste bud, which then essentially opens ion channels, just like in olfaction. Sodium flows into the cell, again making the space less negative and triggering that depolarization, which then triggers the action potential, sending that signal to the brain that salt has been ingested and that is what is being tasted. With type 2 receptors, these particular receptors respond to a different category of tastants, so sweet tastes, bitter tastes, and umami. In this case, the transduction is a little different. So these tastins connect with a protein on the taste receptor cell. These connections activate the G protein, but this one in particular is called gusteucin. That triggering of the G protein opens up those ion channels, calcium flows into the cell, triggering depolarization. This depolarization actually causes ATP to be released from the cell and communicate with receptors on the cranial nerve responsible for our sense of taste, which then allows that signal to reach the brain. Type 3 receptors are what code for sour tastins. In this case, it is actually hydrogen ions that flow into the cell. From there, research shows that the pathway is unclear. It's assumed that hydrogen movement depolarizes the cell membrane and triggers serotonin release. So this particular neurotransmitter is released from the cell and communicates with the sensory neuron to help that signal travel to the brain. This wraps up my discussion of chemoreception. Thanks for listening.